Welcome to this comprehensive review of the physiology of muscle contraction. In this session, we will explore the sliding filament theory, one of the most fundamental concepts in physiology, from the neuromuscular junction all the way to cross the bridge cycling and muscle shortening. Muscle contraction is the process by which chemical energy is converted into mechanical force. Every movement in the human body, from blinking and breathing to walking and lifting, depends on this coordinated mechanism. On the left side of the slide, we can see the anatomy of the calf muscles. The gastrocnemius is the superficial muscle, while the soleus lies deeper beneath it. Together, these muscles insert into the Achilles tendon and generate plantar flexion of the foot, the movement used during the standing, walking, running, and jumping. If we zoom further inwards, we move from the whole muscle to muscle fibers and finally to myofibrils, the microscopic control units responsible for the force generation. Inside these myofibrils are the repeating functional units called the sarcomeres. The sarcomere is the basic contractile unit of the skeletal muscle. It contains two major protein filaments, thin filaments made of actin and thick filaments composed of myosin. According to the sliding filament theory, muscles contract not because the filaments shorten, but because actin filaments slide over the myosin filaments, bringing the Z lines closer together and shortening the sarcomere. Now let's examine the molecular sequence shown on the right side of the slide. The process begins when the calcium ions are released from the sarcoplasmic reticulum. Calcium binds to the troponin on the actin filament, causing tropomyosin to shift away and expose the myosin binding sites on actin. This allows the energized myosin head to attach to the actin, forming a cross bridge. Next comes the power stroke. The myosin head pivots, pulling the actin filament inwards towards the center of the sarcomere. During this step, ADP and inorganic phosphate are released, and mechanical force is generated. After the power stroke, a new ATP molecule binds to the myosin head, causing the detachment of the cross bridge. ATP is then hydrolyzed into ADP and phosphate, re-energizing the myosin head and preparing it for another cycle. This repetitive process is called cross-bridge cycling. As long as the calcium and the ATP remain available, the cycle continues, leading to sustained muscle contraction. The lower panel summarizes the entire sequence, starting from the acetylcholine release at the neuromuscular junction, followed by the action potential propagation through T tubules, calcium release, troponin activation, cross-bridge cycling, muscle shortening, and finally the calcium reuptake resulting in relaxation. Two key requirements are essential for the muscle contraction: calcium, which exposes the binding site, and ATP, which powers the cycle and allows the myosin detachment. Without ATP, myosin cannot detach from the actin, resulting in sustained rigidity, a phenomenon observed in rigor mortis. This elegant physiological process demonstrates how microscopic molecular interactions generate powerful macroscopic movement throughout the human body. Let us apply the physiology of muscle contraction to a real clinical scenario. The case is titled the 400-meter sprint collapse. Our patient is Vikram Nair, a 19-year BSc student and a competitive sprinter. During a 400-meter sprint race, he suddenly develops a severe calf muscle cramp. An important clue in the history is that he skipped breakfast and consumed only water before the event. Soon after sprinting, his calf muscles become rock hard, painful, and locked in contraction. He is unable to relax the muscle. The cramp slowly resolves only after rest and the administration of glucose containing the fluids. So, what exactly happened? What is the physiology? To understand this, we must revisit the role of ATP in muscle contraction. During intense exercise such as sprinting, skeletal muscles require enormous amount of ATP. ATP is essential not only for generating the force but also for muscle relaxation. Many students believe ATP only causes contraction, but in reality, ATP is critically required for detachment of myosin from the actin. In the cross-bridge cycle, myosin binds to actin and performs the power stroke, pulling the thin filament inwards. After this movement, a fresh ATP molecule binds to the myosin head to break the actin myosin bond. Without ATP, the cross bridge cannot detach. As the ATP stores become depleted, myosin heads remain tightly attached to the actin, resulting in sustained contraction and muscle rigidity. This is exactly what occurs in severe muscle cramps. In Vikram's case, several factors contributed to the rapid ATP depletion. First, the 400-meter sprint is an intense anaerobic activity requiring immediate energy production. Second, he skipped breakfast, meaning the muscle glycogen reserves were already reduced. Third, he consumed only water without glucose or electrolyte replenishment. As energy demand exceeded ATP production, cross bridge cycling became impaired. The muscle fibers entered a locked, tense state, producing the painful cramp illustrated on the right side of the slide. The highlighted muscle bundle demonstrates rigid, sustained contraction caused by the energy depletion. This phenomena is closely related to the rigor mortis, where ATP depletion after that prevents myosin detachment, leading to the muscle stiffness. However, in living muscle, rest and glucose administration restore ATP production, allowing the relaxation to gradually occur. This case beautifully demonstrates a key physiological principle. ATP is necessary both for muscle contraction and for muscle relaxation. Without ATP, muscles cannot let go. The core mechanism sliding, not folding. Now that we understand that the clinical importance of ATP in muscle function, let us explore the fundamental principle behind the muscle contraction itself. This slide highlights the core concept of sliding filament theory. For many years, scientists believed that the muscles contracted by folding or collapsing inwards, similar to an accordion. However, in 1950s, electron microscopic completely changed this understanding. Researchers observed that the individual protein filaments inside the muscle fibers did not shorten during the contraction. Instead, the filaments maintained their original length and simply moved past one another. This discovery became the foundation of the sliding filament theory. On the left side of the slide, the red cross presents the old incorrect theory, the idea that muscle proteins physically folded or compressed. On the right side, the green check mark represents the correct mechanism. During contraction, the thick filaments, which are composed of myosin, slide past the thin filaments, which is mainly composed of actin. Importantly, the length of both actin and the myosin filaments remains unchanged. Only the degree of overlap between them increases. As the thin filaments slide inside towards the center of the sarcomere, the entire muscle fiber shortens, producing contraction and force generation. This movement occurs because myosin heads repeatedly bind to actin, performs power strokes, detach using the ATP, and then reattach further along the filament. This repeated interaction is known as cross-bridge cycling. A very important exam concept is this: muscle contraction occurs by filament sliding, not filament shortening. The proteins themselves do not shrink. Instead, the sarcomere shortens because the filaments overlap more closely. This explains several structural changes seen during the contraction. The I band and the edge zone become small, while the A band remains constant because the thick filament length never changes. Understanding this principle is essential for the physiology, sports medicine, neuromuscular disorders, and pharmacology involving the muscle relaxants and anesthetic agents. Targeted neural delivery. Now that we understand how muscle fibers contract, the next question is: how does the nervous system selectively control the muscle movement? This slide introduces the concept of targeted neural delivery through the motor unit and the neuromuscular junction. Let us begin with the key concept shown on the left side. A single motor neuron does not stimulate the entire muscle. Instead, each motor neuron controls only a specific group of muscle fibers. This arrangement allows the body to produce highly precise and coordinated movements. The combination of one motor neuron and all the muscle fibers it innervates is called a motor unit. This is extremely important physiological definition. One neuron plus its designated muscle fibers equals to one motor unit. In the diagram, the blue branching structure represents the motor neuron axon. As the axon enters the muscle, it divides into multiple terminal branches. Each branch forms a connection with a specific muscle fiber. These connections are called the neuromuscular junctions or NMJs. The neuromuscular junction is also known as the motor end plate. It is the precise site where the neuron communicates with the muscle fiber. When an action potential travels down the motor neuron, it reaches the axon terminal and triggers the release of the acetylcholine. Acetylcholine crosses the synaptic cleft and binds to the nicotinic receptors on the muscle membrane. So, this generates the depolarization of the muscle fiber membrane, leading to muscle action potential formation. That signal spreads through the sarcolemma and the T tubules, ultimately triggering the calcium release from the sarcoplasmic reticulum. And once the calcium is released, cross-bridge cycling and the muscle contraction begins. The organization of the motor units also determines the movement precision. Small motor units, such as those controlling the eye muscles or the finger muscles, contain very few muscle fibers per neuron, so allowing the extremely fine control. Large motor units, such as those in the quadriceps or the gastrocnemius, contain hundreds of fibers per neuron, producing powerful but less precise movements. Another important principle is motor unit recruitment. As the demand increases, the nerve system activates additional motor units. This allows the muscles to generate graded contractions instead of an all or none response. Clinically, the neuromuscular junction is highly important because many diseases and drugs act at this site. For example, myasthenia gravis reduces the acetylcholine receptor function, causing muscle weakness. Botulinum toxin blocks the acetylcholine release, leading to paralysis. Neuromuscular blocking agents used during the anesthesia also act here. So, this slide forms the bridge between the neurophysiology and the muscle physiology. The nervous system precisely delivers the electrical signals, and the muscle converts those signals into the mechanical force. The molecular engine, locks, keys, and fuel. Now we arrive at the molecular heart of muscle contraction. This slide explains the interaction between three clinical components which are critical: calcium, troponin and tropomyosin, and ATP. Together, they function like a biological lock, key, and fuel system. Let us begin with the resting state of the muscle. At rest, the actin filament contains special binding sites where the myosin heads could attach. However, these sites are physically blocked by a protein called tropomyosin. As shown on the slide, tropomyosin acts like a safety cable or blocker, a line across the actin filaments and preventing the unwanted contraction. So, this ensures muscles do not contract continuously. Attached to the tropomyosin is another protein complex called troponin. Troponin acts as the sensor of the system. It constantly waits for the signal that contraction should begin. That signal is calcium. Calcium ions or Ca2 plus serve as the molecular key. Under resting conditions, calcium is stored inside the sarcoplasmic reticulum, which acts as the intracellular calcium reservoir. When a muscle action potential travels through the T tubules, the sarcoplasmic reticulum releases calcium into the cytoplasm. The released calcium then binds to the troponin. This binding changes the shape of the troponin complex. As troponin shifts, it pulls the tropomyosin away from the actin binding sites. The lock is now open. The previously hidden active sites on actin become exposed, allowing the myosin heads to attach. This initiates cross-bridge formation and contraction. But contraction cannot continue without energy. That energy comes from the ATP, the fuel of the molecular engine. ATP performs the multiple essential functions. First, ATP binds to the myosin head, allowing the detachment from the actin after the power stroke. Second, ATP is hydrolyzed by the ADP and the inorganic phosphate, which recogs the myosin head into its energized position. Third, ATP powers calcium pumps that return calcium back into the sarcoplasmic reticulum during the relaxation. So, ATP is necessary both for contraction and for relaxation. Without ATP, myosin remains attached to the actin, leading to the sustained rigidity as seen in rigor mortis. This slide beautifully demonstrates the coordination of the molecular physiology. Troponin is the sensor, tropomyosin is the blocker, calcium is the key, and ATP is the fuel. Together, they regulate whether a muscle remains relaxed or contracts forcefully. Clinically, many important drugs act on this pathway. For example, calcium channel abnormalities affect the contraction strength, while malignant hyperthermia involves uncontrolled calcium release from the sarcoplasmic reticulum. Understanding these molecular interactions is therefore essential not only in physiology but also in anesthesia, cardiology, sports medicine, and neuromuscular disease management. Initiating the cross-bridge cycle. Now we walk step by step through the initiation of the cross-bridge cycle, the central mechanism responsible for the skeletal muscle contraction. This process begins with the neural stimulation. On the left side of the slide, we see the sarcoplasmic reticulum or SR, which functions as the calcium storage system of the muscle cell. When a motor nerve impulse reaches the muscle fiber, an action potential travels along the sarcolemma and rapidly spreads deep into the cell through the T tubules. This electrical signal stimulates the sarcoplasmic reticulum to release a large amount of calcium ions into the cytoplasm. Notice the green calcium ions flooding out the SR. This sudden rise in the intracellular calcium concentration is the trigger that activates the contraction. Now, let us move to the right side of the slide where the actual cross-bridge cycle begins. The released calcium ions binds to the troponin, which acts as the regulatory sensor protein. This binding changes the shape of the troponin complex. As the troponin changes the position, it pulls tropomyosin away from the actin filament. At rest, tropomyosin blocks the active binding sites of actin. But once the calcium binds, those binding sites become exposed. This is the critical activation step. Now the energized myosin heads, which already contain stored energy from the ATP hydrolysis, can attach firmly to the actin. This attachment forms the actin myosin cross bridge. Once attached, the myosin head pivots inwards, pulling the thin filament towards the center of the sarcomere. This movement is called the power stroke. During the power stroke, ADP and inorganic phosphate are released and the mechanical force is generated. Immediately after the power stroke, a fresh ATP molecule binds to the myosin head. This ATP binding causes the detachment of the myosin from the actin. Then the ATP is hydrolyzed again, re-energizing the myosin head for the another cycle. As long as the calcium and the ATP remain available, this process repeats continuously, producing sustained muscle contraction. This sequence is known as the excitation-contraction coupling because an electrical signal is converted into the mechanical movement. A very important physiological principle is demonstrated here. Calcium initiates the contraction while ATP powers the cycle. Without calcium, binding sites remain blocked. Without ATP, myosin cannot detach, leading to the rigidity. Clinically, many disorders affect this pathway. For example, hypocalcemia reduces the contraction strength, while ATP depletion contributes to the muscle fatigue and rigor states. This slide represents one of the most fundamental processes in human biology or physiology, that the conversion of an electrical nerve impulse is into the coordinated muscular movement. Recharging and relaxation. After understanding how muscle contraction begins, we now move to the equally important process of muscle relaxation. This slide focuses on two key phases, recharging and relaxation. Let us begin with the left side of the slide, the recharging phase. During sustained contraction, the cross-bridge cycle repeats continuously. Myosin heads repeatedly attached to the actin perform the power stroke, detach and reattach again. For this repetitive cycle to continue, ATP must be constantly regenerated. Notice the ATP symbol shown beneath each myosin head. Each cycle requires fresh ATP. ATP binds to the myosin, causes detachment from the actin, and then undergoes hydrolysis to re energize the myosin head. As long as both calcium ions and the ATP remain available, muscle contraction continues. This is why active muscle consume enormous amounts of energy. Muscle tissue has one of the highest metabolic demands in the body during exercise. Now let us move to the right side of this slide, the relaxation phase. Relaxation begins when the nerve signal stops. Once the neural stimulation ceases, the sarcoplasmic reticulum actively pumps the calcium ions back into storage. This process uses the ATP dependent calcium pumps, known as the SERCA pumps. As the calcium levels in the cytoplasm fall, calcium reassociates from the troponin. Without calcium bond, troponin returns it to the resting shape. This allows the tropomyosin to slide back over the actin binding sites, blocking the interaction with the myosin once again. As the binding site closes, cross-bridge formation stops and the muscle fibers relaxes. One of the most important concepts on this slide is highlighted in the yellow box below. Relaxation is an active process. Many students assume that muscles relax passively, but physiologically, relaxation requires ATP. ATP is needed to detach myosin from actin and pump calcium back into the sarcoplasmic reticulum. Without ATP, the cross bridge cannot break. The muscles remains rigid and locked. This is what exactly occurs in the rigor mortis after death. When ATP production completely stops, the myosin head stays permanently attached to the actin, causing the stiffness of the skeletal muscle. Clinically, defects in the calcium reuptake can also impair the relaxation. For example, certain muscle disorders and malignant hyperthermia involve abnormal calcium handling, leading to prolonged contraction and muscle rigidity. So, this slide teaches a critical physiological truth. Contraction requires calcium, but relaxation also requires energy. ATP is essential not only for the movement but also for stopping the movement as well. The contraction control board. This slide brings together everything we have learned so far into one integrated control system for muscle contraction. Think of a skeletal muscle like a highly advanced biological machine. For contraction to occur successfully, three absolute requirements must be switched on. At the same time, multiple override systems exist to stop the contraction when necessary. Let us first examine the three essential requirements shown on the left side of the slide. The first requirement is neural stimulus. A muscle cannot contract unless it receives an action potential from a motor neuron. The nervous system initiates the entire process. Without electrical stimulation, the muscle remains inactive. The second requirement is calcium. Once the nerve impulse arrives, calcium must be released into the sarcoplasm for the sarcoplasmic reticulum. Calcium acts as an activation signal. It binds to the troponin, moves tropomyosin aside, and exposes the actin binding sites. Without calcium, cross bridge formation cannot begin. The third requirement is ATP. ATP supplies the energy needed for the cross bridge cycling. It powers the myosin movement, enables detachment from actin, and supports the calcium reuptake during relaxation. Without ATP, the contraction system fails completely. So remember this key principle: neural signal, calcium, and the ATP are the three absolute requirements for the muscle contraction. Now let us move to the right side of the slide, which shows four system overrides, the physiological STOP stop mechanisms. The first override energy is system fatigue. If ATP stores become severely depleted, the muscle can no longer sustain contraction. This commonly occurs during prolonged intense exercise. The second override is nervous system fatigue. Over time, motor neuron signaling may slow down or fail, reducing the stimulation to the muscle. This contributes to the central fatigue during exhaustive activity. The third override is voluntary control. The brain can intentionally stop sending signals into the skeletal muscles. This is how conscious movement is controlled. Unlike cardiac muscle, skeletal muscle contraction is under voluntary somatic control. The fourth override is the sensory reflex system. Pain receptors, tendon organs, and stretch receptors continuously monitor the muscle tension and injury risk. If excessive force or damage is detected, protective reflexes can abruptly inhibit the contraction. A classic example is the Golgi tendon reflex, which prevents the tendon rupture during excessive load. This slide demonstrates that the muscle contraction is not simply a mechanical movement, it is a tightly regulated physiological process requiring coordination between the nervous system, energy metabolism, ion signaling, and the protective reflex pathways. Clinically, disruption of any one of these systems can produce muscle weakness, fatigue, spasm, or paralysis. For example, electrolyte imbalance affects calcium signaling, mitochondrial disorders reduce the ATP generation, and motor neuron disease impairs neural stimulation. So, the slide acts as the master control summary for everything required to produce safe and effective muscle contraction. Decoding Vikram's rigid calf. Now we return to Vikram's clinical case. The 19-year-old sprint athlete who suddenly developed a rigid calf muscle during a 400-meter race. At first glance, it may appear to be a simple muscle cramp. But when we apply the physiology we just learned, the real mechanism becomes very clear. This slide explains exactly why Vikram's muscle became locked in contraction. Let us begin with the diagnosis shown on the left side. Vikram sprinted intensely on an empty stomach. He skipped breakfast and consumed only water before the race. During high intensity sprinting, skeletal muscle requires enormous amounts of ATP. ATP production depends heavily on the available glucose. Because his glycogen and the glucose reverses were insufficient, his muscle rapidly exhausted its ATP supply. This is the key event in the entire case. Now, look at the center section, the mechanism. Remember the critical physiological rule from the previous slide. ATP is required not only for contraction but also for relaxation. Many students incorrectly think that ATP only creates movement. In reality, ATP is essential for breaking the actin myosin cross bridge. Without ATP, the myosin heads cannot detach from the actin. As ATP levels fell dramatically in Vikram's muscle, the cross-bridge cycle became trapped. The myosin heads remained tightly bound to the actin filaments. The muscle fibers became locked in sustained contracted state. This is physiologically similar to the rigor mortis, where the ATP depletion after death causes permanent muscle stiffness. Now observe the right side of the slide. The highlighted warning, critical ATP depleted, represents the moment the muscle lost its ability to relax. The result was a rigid, painful, tense calf muscle. Importantly, the muscle did not relax immediately with stretching alone. Why? Because the underlying problem was biochemical, not mechanical. The muscle required restoration of ATP production. That is why the cramp slowly resolved only after rest and IV glucose administration. The glucose restored cellular energy metabolism, ATP synthesis reassumed, and the myosin heads detached. Also, the relaxation finally occurred. This case beautifully demonstrates one of the most important concepts in muscle physiology. Relaxation is an active energy dependent process. Clinically, similar mechanisms can occur in severe exercise exhaustion, electrolyte imbalance, ischemia, heart related or heat related illness, and metabolic disorders affecting the ATP production. This slide also explains why hydration alone is not always enough in athletic cramps. If energy stores are depleted, the real treatment is a restoration of the metabolic fuel. So, the final takeaway from Vikram's case is simple: no glucose means no ATP, no ATP means no cross bridge release, and no release means persistent muscle contraction. That is the physiology explanation behind his rigid calf. Pathology maps to molecular failure. Now that we understand the normal physiology of muscle contraction and relaxation, we can connect these mechanisms directly to the clinical disease. This slide is extremely important because it shows how different pathologies are actually failures of same molecular system. Every disorder in this table affects one of the major control points we previously discussed: ATP, calcium, or neural stimulation. Let us analyze each condition one by one. First, rigor mortis. It occurs when the death ATP production completely stops. Without ATP, the myosin heads cannot detach from the actin filaments. As a result, the actin myosin cross bridges become permanently locked. Relaxation fails completely. This produces the characteristic post mortem muscle stiffness. So, rigor mortis is essentially a state of irreversible contraction caused by total ATP depletion. This is the same mechanism we saw in Vikram's severe sprint cramp. But in rigor mortis, ATP can never be restored. Next, malignant hyperthermia. This is a life threatening pharmacogenetic disorder often triggered by anesthetic agents such as succinylcholine or volatile inhalational anesthetics. The primary defect is controlled and then uncontrolled when the calcium release from the sarcoplasmic reticulum is more. Massive amount of calcium continuously floods in the muscle cell. Remember, calcium keeps the contraction machinery switched on. Because calcium release never stops, the muscle enters the sustained contraction. This dramatically increases the ATP consumption and heat production, leading to severe hyperthermia, muscle rigidity, acidosis, and rhabdomyolysis. The treatment is dantrolene. It works by blocking the calcium release from the sarcoplasmic reticulum, thereby inhibiting the contraction cycle. This is a classic example of targeting molecular physiology to save a patient's life. Now let us examine severe muscle cramps. Here, the major issue is usually fatigue, dehydration, electrolyte imbalance, or temporary ATP depletion. In these situations, calcium is not fully pumped back into the sarcoplasmic reticulum. As a result, partial contraction continues even when the muscle should relax. This creates painful, involuntary tightening of the muscle. Unlike rigor mortis, the process is reversible once the ATP, electrolytes, and hydration are restored. Finally, organophosphate poisoning. This condition involves excessive acetylcholine accumulation at the neuromuscular junction. Normally, acetylcholine is rapidly degraded by the acetylcholinesterase, but organophosphates inhibit this enzyme. As acetylcholine accumulates, the muscle receives continuous neural stimulation. The motor end plate remains constantly activated. This produces sustained fasciculations, muscle twitching, spasm, and eventually paralysis due to the receptor exhaustion. Clinically, patients may also develop respiratory failure because the diaphragm becomes affected. So, despite appearing as complete different diseases, all four conditions are actually failures of the same contraction relaxation system. Rigor mortis represents the ATP failure, malignant hyperthermia represents the uncontrolled calcium signaling, severe cramps represent incomplete calcium recovery and energy depletion, organophosphate poisoning represents the uncontrolled neural stimulation. This is why understanding basic physiology is so powerful. Once you understand the molecular machinery, clinical pathology becomes much easier to decode. And that is the central lesson of this slide. Every muscle disorder is ultimately a problem of energy, calcium, or neural control. Now, let us finish with a high yield board style review question, which is commonly asked in MBBS, PharmD, NCLEX, USMLE, and other medical entrance examinations. The question asks which protein directly blocks the myosin attachment site on actin during the resting state. The correct answer is tropomyosin. This is one of the most commonly tested concepts in muscle physiology. Many students incorrectly choose troponin, but remember the distinction carefully. Troponin is the sensor protein. Its role is to detect calcium. When the calcium binds to troponin, it triggers a conformational change. The conformational shift pulls tropomyosin away from the active binding sites on actin. So, tropomyosin is the actual physical blocker. Think of tropomyosin as a protective cable lying across the actin binding sites. At rest, it prevents myosin heads from attaching. Once the calcium enters the sarcoplasm, troponin activates and moves the blocker aside. Only then cross-bridge formation can occur. This is why the phrase troponin senses and tropomyosin blocks is extremely useful in exams. Now let us review the additional high yield fact at the bottom of the slide. During muscle contraction, the filaments slide past each other but they do not change the length. As a result, the A band remains constant because it presents the full length of the thick myosin filament. Meanwhile, the I band shrinks because the thin and the thick filaments overlap more during the contraction. The edge zone also shrinks and the actin filaments move closer towards the center of the sarcomere. This concept is frequently tested in physiological practicals and competitive exams. So remember the golden rule. A band stays the same, I band and edge zone decreases during contraction. And with that, we complete the molecular physiology of muscle contraction. From neural stimulation to calcium signaling to cross-bridge cycling, clinical pathology and board review integration. Understanding these mechanisms does does not just help in exams, it helps in real clinical disease at the molecular level. Core takeaways and next steps. Now, let us conclude the lecture with the most important concepts that you must remember from the muscle contraction physiology. These are the core takeaways that connect every slide we discussed today. First, muscle filament slide. They do not fold. This is the foundation of the sliding filament theory. During contraction, the thick and the thin filaments move past each other while their actual lengths remain unchanged. Second, no calcium means no contraction. Calcium is the activation key. Without calcium release from the sarcoplasmic reticulum, troponin cannot activate. Tropomyosin continues blocking the binding sites, and cross-bridge formation cannot occur. Third, no ATP means no relaxation. This is one of the most clinically important concepts in physiology. ATP is not only required to just generate the force, it is required to break the actin myosin connection. Without ATP, the muscle remains locked in contraction. That is exactly what happens in rigor mortis, extreme fatigue, and severe ATP depletion states. Now look at the central summary box. Think of muscle physiology using these three simple analogies. Calcium is the key, ATP is the fuel, and tropomyosin is the lock. If the key is absent, the system never activates. If the fuel is absent, the system cannot reset. And if the lock remains closed, myosin cannot bind. These three components together control every skeletal muscle movement in the human body. Now let us move to the audience challenge on the right side. The question asks if you had to lose one, which is more critical for survival? ATP or calcium. This is designed to make you think physiologically. Without calcium, muscles cannot contract properly, but without ATP, cells cannot survive at all. ATP powers the ion pumps, membrane stability, neural conduction, active transport, and virtually every cellular processes in the body. So, although both are essential, ATP is ultimately the more fundamental survival molecule. Finally, the next lecture will expand this foundation even further. We will move into the neuromuscular junction physiology, sarcomere structure, A bands and I bands, and important clinical disorders such as Lambert-Eaton syndrome. So, this lecture was not just about memorizing the contraction concepts. It was about understanding how muscular physiology explains real movement, real pathology, and real clinical medicine. Once you truly understand the mechanism, you are no longer need to memorize the isolated facts because the physiology itself gives you the answer. Thank you for watching this lecture till the end. I hope you understood all the concepts which were required for understanding the muscle contraction, not just for the exams but clinically and conceptually as well. If you found this video helpful, don't forget to like, share, and subscribe for more high yield medical lectures, clinical case discussions, and board review content. Drop your answers and doubts in the comment section below. I would love to hear your thoughts. Until next step, keep learning, keep questioning, and keep saving lives. Thank you, and see you in the next lecture.